Hi, I'll be talking about molecular basis of inheritance. We had looked at chromosomal level of inheritance in chapter 15. And so this is chapter 16, molecular level of inheritance. So we'll be looking at the pattern of inheritance up to DNA level, like on the molecular level. All right. So I will go ahead, few things. I will focus on few things that I felt that they are very important to note there. Okay, so in the previous chapter, we emphasized that part of the things that Watson discovered, or that was emphasized by Mendel rather, was that the part of the body that is responsible for inheritance is the gene, so that the gene is what is responsible to transmit traits. Okay, so here we'll be looking at the DNA level now. Okay, so this is what the DNA looks like. It's an helical shape. We say it's a double helical shape. So DNA is called deoxyribonucleic acid, as you can see here, and that was what proposed by Watson and Crick in the year 1953. All right, less of this history now. We are going to, so now we want to look at the fact that the DNA is actually the genetic, the content in the gene that is responsible for trait transmission. All right, so this is what DNA replication looks like. We are going to look at that. We say that DNA replication is semi-conservative, and I'm going to explain that to you as we go. Semi-conservative means half of the DNA is conserved, is preserved. Half of the original DNA is preserved in the replication of the new one. I will show you that as we go. Now, so the emphasis is that DNA is the genetic material. In the early 20th century, the identification of molecules of inheritance posed a major challenge in biologists to the biologists. Morgan showed that genes are located on chromosome, like we emphasized in chapter 15, that two components of chromosome, the DNA and protein. I told you that DNA is wound around by histone protein, all right? And I told you that when DNA and histone protein are condensed, we call it chromosome. When they are uncondensed, we call it chromatin. All right, so the role of DNA in inheritance was first discovered by studying bacteria and viruses that infect them. Let's go. Okay, now what majorly they did was that they used the concept that we call transformation. So the discovery of genetic material of DNA began with the research that Griffith did in 1928. He worked with two strains of bacteria. One is pathogenic, it can cause infection. The other is harmless. So he mixed the heat killed the remain of the pathogenic strain that has been killed by heat. He mixed it with the living cell of the one that is harmless, that is not pathogenic. Now, it discovered that some of the living cells now became pathogenic by mixing the remains of the dead pathogenic ones with the living one. Then over time, the living one became pathogenic. And so what was termed is transformation. Now, and that's defined by a change in genotype and phenotype due to assimilation of foreign DNA. I'll talk more about that. Okay, let's go. Now, in that context, that is what we call it transformation. And for some of you, you've done the gene transfer, right? When we talk about how an organism can pick up the genetic component of other organisms, and then they pick it up and become part of them. It gets inserted into their DNA, into their gene, and then begins to replicate. And so it changed the configuration of such organisms. That is what we call transformation. All right, that's transformation. So more evidence for DNA as genetic. All right, so more evidences for DNA as the genetic material came when they also studied virus that infects bacteria. So they use some virus that are called bacteriophage or phages. And so the virus is DNA, sometimes like RNA, right? Enclosed by protective coats, often simply protein. So the phages have been widely used as tools by researchers in molecular genetics. I'm just going to run through that. So that is what the phage looks like, and that is the phage going to infect the bacteria cell. Okay. Now, Ashe and Chase showed in 1952 that the DNA is the genetic material of a phage known as T2. They designed an experiment. Then with that, they also concluded that injecting the DNA of a phage into the bacteria is going to cause the bacteria to take up that DNA and then show the phenotypic relationship that stem from that DNA of that virus as well. All right. Now, so I'll start to talk about this now. I just want to run through that. So what is a DNA? The DNA is simply, first is called deoxyribonucleotide, and it's a group of nucleotides. Now, what is a nucleotide? Nucleotide consists of three major things. There are three major components of a nucleotide. Number one, nucleotide is made up of nitrogenous base, and the nitrogenous bases are adenine, thymine, guanine, or cytosine. You'll be talking about uracil when we get to the RNA, but we are not there yet. So those are the four nitrogenous bases that are present in DNA. So nitrogenous base number one, sugar number two, and phosphate group number three. Those are the three components that makes up a nucleotide. So in 1950, Chagav reported that DNA composition varies. All right, so Chagav came up with a rule that thymine is going to pair with adenine, and guanine is going to pair with cytosine. So he came up with the rule ATGC. So A pairs with T, G pairs with C. So that's the rule that Chagav came up with, GC. So anywhere you see A, it goes with T. Anywhere you see T, G, it goes with C. All right. So that was Chagav rule. The two findings became known as Chagav rule, that the base composition of DNA varies between species, and that in any species, A and T bases are equal, and G and C also goes together. And so the basis of the rule ended up giving the DNA a double helical shape. All right. So this is what Chagav came up with. If you look at this, you will see that you will see how the DNA is oriented, and this is how the this is here every so take note every DNA, like I said, they are made up of nucleotides. First is the nitrogenous base, second is the phosphate, and third is the so you have the nitrogenous base, you have phosphate, and then you have the sugar. So you see the nitrogenous base is here, the phosphate is here, and here is the sugar. So those are the three structures that makes the, all right? So those are the three components that makes up a nucleotide: the phosphate group, the sugar component, and the nitrogenous base. Now, so look at this now. This is the phosphate. Here is the phosphate here. Here is the sugar. We call it a 5-carbon sugar, and then here is the nitrogenous base. So in nitrogenous base, so this is like half now. This is going to now pair up again with another for the double strand, which you're going to see as we go. All right, so the DNA was accepted as the genetic material, and now the issue was the structure, which is what uh, Franklin came up to design, which we're going to look at now. So the structure of DNA is double helix, and so Franklin X-ray crystallographic images of DNA allowed James Watson to deduce that DNA was helical, like it has the helical shape. The X-ray images also enable Watson to deduce the width of the helix and the spacing of the nitrogenous base. The pattern in the photo suggested that DNA molecule was made up of two strands, key thing, two strands, and that it formed a double helix. It is coiled on each other, two strands that are wound on each other. That is what it means by double helix. So Watson and Crick built model of a DNA double helix to conform to the X-ray that Franklin has came up with, and Franklin then concluded that there were two alpha sugar phosphate backbone. So the sugar phosphate is the backbone of the DNA. I'll show you I'll show that in a moment, with the nitrogenous bases acting as the attachments or side chains. So Watson built a model in which the backbones were anti-parallel, their subunits run in the opposite direction. I'll show you that. So at first, so look at this. So this is a view of what Watson had said, which I'm going to go back to continue the slide, but look at this. So Watson concluded that DNA runs from 5' to 3', and that another strand runs from 3' to 5'. So in other words, they say that it is in the opposite, both strands run in the opposite direction, as though you have this and then this way. So that the two strands were running in opposite directions, so we say that it is anti-parallel to each other. Again, there are two strands. The backbone is the sugar phosphate, and is the sugar and phosphate makes the backbone, make the structure, and the nitrogenous bases form the attachment, the side chains, and that the strands were placed in opposite direction, anti-parallel to each other. Just like you can see here, one start from 5' to 3', the other from 3' to 5'. So it's like head to toe, toe to head. So that's what they came up with the arrangement of the strands. Okay, so at first, all right, so you see Watson built a model in which the backbone were anti-parallel to each other. So at first, Watson and Crick thought the bases paired like with like A and A to A, but later they found out that it's actually the alternate, that it's A pairs with T and G pairs with C. Now, what they discovered is this: purines, that the bases are different. The two that ends with NINE, 9, they are purines. So purines are A and G. Adenine and guanine are purines. So if you look at this, these are purines. Adenine and guanine are purines. Pyrimidines are cytosine, and thymine. So you see that the ones that ends with the purines ends with 9. Adenine, guanine, they ends with 9. All right, let's go. All right, so the Chargaff rule is what states and emphasizes that G, the pairs with C, and C pairs and A pairs with T. Like I told you, the purines are the ones that end with 9. So adenine and guanine purines, then they're going to pair with the pyrimidines. All right. So that's what you have like this. So you see, this is a purine. Purines again are double bonds. I mean, they are double rings. So you see, this is a double ring, this is a purine, another double ring, a purine. They pair with single rings, which is thymine and cytosine. All right. So the next thing we'll be looking at is DNA replication. How does DNA replicate? All right. If it's the molecule that is responsible for inheritance, how do they also reproduce? How do they replicate? That's what we've been checking now. So the resemblance of offspring to parent relies on accurate replication of DNA prior to meiosis. You of course you still remember meiosis, right? And if you remember, in meiosis or mitosis, we talked about interphase. That at the interphase there is G1, growth phase 1, there is replication phase, which is the S phase, and there is the G2. So the S phase is the DNA replication phase. So what we want to check now is the details of the DNA replication. What actually happened when we say the DNA is replicating? All right, so replication prior to meiosis ensures the faithful transmission of genetic information from a parent to the daughter cell. So if any replication happened before meiosis happen, anything that happened before meiosis ensures that there is going to be the transmission of that gene or that DNA to the offspring. So Watson and Crick noted that a specific base pairing suggested a possible copying mechanism for genetic material, and that is what we call DNA replication. So I'm going to show you what is the secret about DNA replication now. Now, I've told you that every DNA strand have double strands. So there are two strands that are present in every DNA strand. Every DNA strand is made up of two strands. But the summary of DNA replication is this. In the parent, there are two strands. In the new strand, there are also two strands. But when a DNA is replicating, as though a mother DNA is giving birth to a child DNA, it is not the DNA that is new, is not entirely new. It is half new. Half the parent, half new. Half the parent, half new. So in essence, this is what is going to happen in DNA replication. A DNA strand is going to give rise to a new DNA strand, but it's going to give rise to two. And so it's going to put, this is what happens actually. I will do a little schematic view. So this is a DNA strand. By the time this DNA strand is going to give rise to two new DNA, one of the new, we have this. The second new, we have this. Then they are going to have a new strand with it. So this and this. So you end up saying that the new strand is actually not 100% new. It is half the parent, half the new. So we say that DNA replication is semi-conservative in that it's going to conserve the parent half. So half of the parent makes the new. So it's semi-conservative. All right, let's go. So that's what we call DNA. Since the two strands of DNA are complementary, each strand acts as a template for building a new strand in the replication. You see that? So one of the strands is acting as a template. This is two exact replica of the parental molecule. All right. So here is what we have. this is one DNA, and this is the DNA strand. You can see that this is the parent strand here, and of course here is the parent strand. The first thing is that they have separated, so each of them will serve as a template for the new one. So by the time this has come here, you see this here. This one is this strand here, and now a new strand was now made to match with this. So you see A pairs with T, A now pairs with T, C pairs with G, T pairs with A pairs with T, G pairs with C. That is the new strand. So this is the new strand now, coupled with the host strand, making the new DNA. And also, this second strand is this. Then this is the new strand. If you look at something critically with me, you will notice that on this new strand, this old strand is 5' to 3'. But this new strand is 3' to 5', so it's anti-parallel to each other. Look at this. This is 3' to 5', and this is 5' to 3', anti-parallel. If they are going to be replicated, will it also be in the same direction? I'll be talking about that. So Watson and Crick concluded that the replication of DNA happened in a semi-conservative model, which is what I've just explained. Right? Again, this is what semi-conservative model means. It means that the host strand will make itself half of itself in the new strand, in the new DNA, and the new DNA is just going to be old plus new. So they also proposed, they looked at the conservative model when it is 100% of the parent, like you can see here, and discovered that that's not the right one, dispersive, in a way that it breaks and form a new one with the hold inserted into it. That's not the one. So what is accepted is the semi-conservative replication. All right. So the copying of DNA is remarkable in its speed and accuracy. More than a dozen enzymes are involved, and the other proteins are also involved in the process. So there are different ways that replication happen in prokaryotic and eukaryotic cells, and I'll be talking about that. So replication begins at the point where we call origin of replication. It's the origination of replication. And so that is the point where the DNA strand will first be opened and be separated. Of course, you see that here. It shows that there will be separation of the strands before DNA replication will begin. But where the strands will begin to separate, we call it origin of replication. All right. Now, look at it here. So for eukaryotic organism, they may have many origin of replication, but prokaryotic only have one origin of replication. Take note of that. Eukaryotic, we have many origin of replication, but prokaryotic cell only have one. And think about it. If you remember, when we're looking at cells, prokaryotic cell only have one circular DNA. So once the origin of replication opens at one point, the DNA will just be replicated throughout run. But for eukaryotic cells, there are many linear DNA. So that means many linear chromosomes. So each of them will have their origin of replication, right? Because there are millions of DNA strands, right? So that is the way it works. So eukaryotic cells have multiple origin of replication, while prokaryotic cells have just one. So you can see this. This is prokaryotic cell showing that just that one origin of replication, and it's going to open here. When this replication bulb opens here, replication then continues there. We call that replication fork. So it continues down, and then the new DNA strand will be made. But in prokaryotic cell, in eukaryotic cell, you're going to see multiple replication origin starting, and so you're going to have multiple strands, multiple origin of replication rather. So I will show you that as we go. Now, at the end of each replication bubble is what we call replication fork. All right, that replication fork is a Y-shaped region where parental DNA strands are being unwound. All right, then there is an enzyme. Because DNA strands are wound together, before they can be separated, there has to be an enzyme that will open them up. So the enzyme that open them, we call it helicases. Helicase is the enzyme that opens them. But before they are opened, the pressure that's holding them together need to be relieved to make it easy to open. So it's more like to prepare them for the untwisting. So the enzyme that does that is the topoisomerase. So you see, it relieves the strain of twisting of the double helix by breaking, swiveling, and rejoining DNA strands. So after that, helicase now come to untwist. Now, to prevent them from coiling back on each other, SSBP, single-stranded binding protein, is going to bind to them so that they don't come back to rejoin. All right, so we call it SSBP. So every part that has been uncoiled will be stabilized by SSBP so that it does not join back to each other. So we call it SSBP, single-stranded binding protein. Let's go. Now, this is what I'm talking about. So you see, the first thing is that topoisomerase will go ahead. So think about let me use my annotation now. Think about it that topoisomerase is coming from this direction and it has gone ahead to relieve the pressure, as though to weaken it, to make it easy to open. Now, helicase is then going to bind and keep opening it. As helicase goes ahead and opening it, of course, these things were coiled on each other. Think about something that is coiled on each other. If you open it, they will likely coil again. So to prevent recoiling, single-stranded binding protein binds on it, and that prevents it from recoiling. The next thing that is going to happen is that now the replication fork has been opened and the DNA is ready to be synthesized. But see, DNA now see, DNA cannot replicate itself first. DNA replication does not begin by making DNA. See you on the next chapter, on the next recording.